Ecco Cinzia, ecco Roberto.
Siamo qui, ti aspettiamo.
Allora, io volevo solo portare i saluti della Società Italiana di Cardiologia e i complimenti a Cinzia e a tutto il gruppo di studio, che è un gruppo di studio molto attivo, forse tra i più attivi, e quindi questo è per noi una fonte di grandissima soddisfazione. E oggi cominciamo proprio con voi il ciclo di webinar della scuola di cardiologia. Abbiamo istituito una scuola di cardiologia che comincerà e che diffonderà tutti questi webinar non solo ai gruppi di studio, ma a tutte le scuole di specializzazione. E di questo ovviamente devo ringraziare il professor Pedrinelli, ma soprattutto il professor Mercuro, che ha fortemente voluto questa iniziativa. Quindi io darei la parola al professor Mercuro e al professor Pedrinelli per i saluti e veramente ancora tanti complimenti e congratulazioni.
Beh, davvero un saluto breve e che non sottragga tempo a questo, diciamo, primo evento di webinar condiviso. Cominciamo da un punto di vista, da un aspetto importantissimo, appunto, le cognizioni basic prodromiche, diciamo, alle conoscenze della fisiopatologia e infine della clinica cardiovascolare. Quindi io mi compiaccio, così come il professor Indolfi, che insieme col quale avevamo proposto in un'epoca forse un po' anticipata questa iniziativa, che ora trova la sua, diciamo, espressione più completa e più strutturata proprio in questo ciclo di webinar. Quindi buon lavoro a tutti e do la parola al chairman della riunione.
Allora, buonasera a tutti, sono Cinzia Perrino, coordinatrice del gruppo di studio di biologia cellulare e molecolare del cuore. Innanzitutto vorrei ringraziare il professore Indolfi per l'energia che profonde quotidianamente nella SIC e nei gruppi di studio, e un ringraziamento particolare va anche ai professori Pedrinelli e Mercuro, che devo dire danno una fonte di ispirazione, un incoraggiamento anche quotidiano. Col professore Pedrinelli ormai ci messaggiamo regolarmente, quindi è veramente un rapporto al di là della professionalità anche umanamente molto importante e piacevole. Il gruppo di studio di biologia cellulare e molecolare del cuore è molto orgoglioso di poter inaugurare i webinar della SIC, sia perché sono diciamo di una grande tradizione e la SIC anche di grande supporto. Infatti ringrazio Alessandra Bazzani e anche l'ingegnere Canino, che è sempre presente per le nostre difficoltà telematiche, eccetera. Questo webinar ha una caratteristica particolare, legata al fatto che noi abbiamo recentemente fatto un sondaggio nei gruppi di studio, e da questo sondaggio è uscito fuori che le aspettative dei soci del gruppo sono proprio quelle di partecipare a un gruppo di studio di una società scientifica per avere un maggiore coinvolgimento nell'attività. E proprio da questo sondaggio è venuto fuori che i webinar possono essere diciamo, una modalità preferenziale per queste modalità di comunicazione. Abbiamo anche fatto una sorta di votazione di vari topics che potevano essere più interessanti, e come dicevo alla professoressa Brundel qualche settimana fa, il topic dell'argomento che verrà trattato oggi, cioè la proteostasis derailment, in generale i meccanismi molecolari della fibrillazione atriale, è stato il topic che ha avuto più voti in assoluto. Questo a indicare come la mancanza di cognizioni o comunque la sete di informazioni per questo tipo di patologie è molto elevata. Just a few words to introduce Professor Brundel. I mean, she's a full professor at the Physiology Department at Amsterdam UMC, University Medical Center. And basically, she was trained both in molecular and cellular biology, but also in clinical pharmacology. And she has been doing basic research on proteostasis to investigate the molecular basis of cardiomyopathies and atrial fibrillation. But the interesting aspect of Professor Brundel's clinical training and also research experience is the ability to combine basic research with a strong translational impact. And I think that's the important thing that inspired this webinar and basically the working group of the cellular and molecular biology of the heart of the Italian Society of Cardiology. So I don't want to spoil anything, and I would go straight to Professor Brundel's presentation.
Thank you, Cynthia. I thank you for your kind introduction. It's a pleasure to give an overview of our research, translational research in atrial fibrillation. I will share my presentation. I think you can see my PowerPoint.
Yes, we do.
Yes, yeah.
Yeah. Okay, so what I will do today is I will give you a few examples of our fundamental research and how we translate the findings to the clinic. You know all about atrial fibrillation. I will not give an introduction on this cardiac arrhythmia, but I want to highlight that atrial fibrillation is characterized by atrial electrical chaos and contractile dysfunction, and this often leads to irregular heart rate. And what is important is that patients have a higher risk to develop blood clots and heart failure, and therefore it needs treatment. So in most patients with AFib, it's a progressive disease. So if you have paroxysmal forms of the disease, often these patients in the end develop the persistent form of the disease. AFib is a serious disease, so it needs treatment, such as ion channel blockers, which are developed in the 60s, or a patient can get an electrical cardioversion to reset the heart rate, or patients are subjected to ablation of certain areas around the pulmonary veins. But unfortunately, all these treatments are not effective to stop AF progression. And often patients have a recurrence of AF within a year. So as a biologist, I'm very much interested in the underlying mechanism in the cardiomyocytes. So what is happening in the atrial cardiomyocyte and in the atrial tissue during AFib? And we already observed a long time ago that persistent AF mainly leads to structural damage in the atrial cardiomyocytes itself. So for example, here you see an EM picture of a patient in persistent AF, and you can clearly see, for example, that the sarcomeres disappeared. And this is called myolysis, so the degradation of the sarcomeres. So you can imagine that if you have structural damage in the atrial cardiomyocytes, ion channel blockers or electrical cardioversion or ablation will not be very effective to treat AF. So we set out to dissect the mechanisms underlying structural damage, and we found that structural damage is rooted in derailed proteostasis. And what is proteostasis? Proteostasis is defined as the balance, the homeostasis in protein expression, function, and clearance. And I will guide you through this definition via this picture, which I adapted from a paper from Hardall in Nature. In a normal situation, proteins are formed in the ribosomes, and with the help of chaperones, especially heat shock proteins, they are folded and traffic throughout the cell. So if you have an expression of an ion channel, for example, the ion channel, with the help of heat shock proteins, the ion channels are expressed or transported to the cell membrane where they can have their function. And what happens during stress, such as atrial fibrillation? The native active proteins can become degraded. Proteins can become misfolded, and if they are misfolded, they tend to form protein aggregates in the cytosol. And with the help of a process called autophagy, these aggregates are cleared out of the cells. And for the autophagy process, the microtubule network is very important. So we observed already many years ago that, for example, chaperones, the heat shock proteins, become exhausted in patients with persistent atrial fibrillation. We also observed that the microtubule network becomes disrupted, and therefore it can hamper cell function. We observed ER stress and excessive activation of autophagy in patients with persistent AF. And last but not least, we observed that patients with AF have increased levels of DNA damage, which of course will influence the cell function. And during my presentation, I will highlight our key findings related to heat shock proteins, the microtubule network, and DNA damage, and how we translate the findings to the clinic. First of all, I briefly explained some model systems we use in our lab to understand the mechanisms underlying AFib. And one of the model systems we use are cultured cardiomyocytes, which we can subject to tachypacing, so rapid pacing. Here you see the contraction of a cell under normal circumstances. But when we subject the cells to rapid pacing, you can clearly see that the green flashing of the cells is reduced. And these movies can be used to make traces, as you can see here below. And we observe that if you tachypace the cells will develop contractile dysfunction in terms of loss of calcium transients and loss of cell shortening, so the actual contraction. We also observed, if you look to the structure, the degradation of the sarcomeric structure. So therefore, this model system shows some endpoints which we also observed in the past in patients with AF. Another model system we introduced in the lab is a tachypaced Drosophila prepupa model. The nice thing of the prepupa of the Drosophila is that they are transparent and are immobile. So when you subject them to tachypacing, they cannot crawl away or fly away. And this gives some advantages. So here you can see a part of, okay, I continue. So here you can see a movie of the heart wall of the Drosophila. Under normal circumstances, it has an irregular heartbeat, as you can see over here. When we subject the prepupa to tachypacing, the heart wall adapts to the high pacing rates, as you can see here in the movie and in the traces on the left. And after tachypacing, this is the same prepupa as the first movie, we see contractile dysfunction. And when we look through the electron microscope to the sarcomeric structure, then we can clearly see already the first steps of myolysis of the sarcomeric structure. And the nice thing of the Drosophila is that you can overexpress or knock out specific genes in the heart, and we successfully used the Drosophila for drug screening. The third model or approach we use in a lab is the use of our biobank. Several years ago already, we started to collect atrial tissue samples, like the right and left atrial appendages of patients with paroxysmal, persistent, long-standing, persistent AF, and also controls in normal sinus rhythm. And we collected human blood samples. In the meantime, we have over 700 samples of these patients. And we used the human biobank to validate our findings from the experimental model systems. So what did we learn in relation to heat shock proteins? Heat shock proteins are cardioprotective proteins. We know already for many years that heat shock proteins can bind to contractile proteins and make them more stable. And in 2006, I already published that heat shock proteins become increased during paroxysmal atrial fibrillation, but become exhausted in patients with persistent AF. And we observed this correlation that patients with a high level of structural damage, especially myolysis, show lower levels of heat shock proteins. And this observation led to the hypothesis that what happens if you increase heat shock proteins? Will this protect against structural damage and AF promotion? So we overexpressed specific heat shock proteins in the HL1 cells by transient transfection methods. And we measured the contractile function. As you can see over here, contractile tachypacing results in contractile dysfunction, but if you overexpress specifically the heat shock protein 27, we observed conservation of contractile function. We tested several heat shock proteins, but especially heat shock protein 27 was highly protective. Heat shock protein 27 also protected against myolysis in cell culture. The same approach was used in a Drosophila prepupa. Here we expressed several small heat shock proteins in the heart of the Drosophila, and especially the heat shock protein 27 protected against tachypacing-induced dysfunction in Drosophila, and it protected against tachypacing-induced structural damage in the Drosophila heart. So these findings were very interesting observations and showed us already that heat shock proteins can have a protective effect. But how can we translate this to the clinic? Here too, we selected a compound from the literature which was known to increase the heat shock protein levels in other organs. And this compound is called geranyl acetone, in short, GGA. And it was already known from the literature that it can protect against neurodegenerative diseases, ischemia, reperfusion in the heart, apoptosis, and against kidney injury. But it was never tested in AFib. So what we did, we treated the HL1 cells with GGA, and you can nicely see the increase of heat shock protein 27 in the atrial cardiomyocytes. And we subjected the cells to tachypacing. And as you can see over here, GGA highly protects against contractile dysfunction in the cells. So this was a very interesting observation, and we observed the same protective effects by GGA also in the Drosophila. But everybody can understand that if you show these data in cells and in Drosophila, the step to the clinic is still a big step. And at that time, before I go to the animal findings, we also showed in this experiment that the protective effect of GGA is via heat shock protein 27, because in this experiment we showed that that siRNA against HSP27 can knock out HSP27 levels in the cells, and when the cells were treated with GGA, the protective effect of GGA was gone. Because if you put a mock siRNA construct in the cells, the protective effect after tachypacing of GGA was still there. So this is just an example how nicely the cells can be used to really mimic the molecular pathway, how a drug can have protective effects against contractile dysfunction. Now the translation more to the clinic. In that time, I worked as a fellow in the Montreal Heart Institute in the lab of Stanley Natel. And probably you know that in this lab, they have a dog model for atrial fibrillation. And in these dogs, received a pacing device in the atria, and the dogs were tachypaced for up to 7 days. And during this process of pacing, you can see that the dogs become more vulnerable to develop spontaneous atrial fibrillation. But when the dogs were pre-treated with GGA, you can clearly see the protection against atrial fibrillation. So this was a very nice proof that GGA protects against tachypacing-induced AFib. Another model in the lab of Natel is the heart failure-induced AFib model. In this model, the ventricles are tachypaced, which results in more atrial fibrillation in the atria. Here, GGA was also highly protective, as you can see. And in this model of heart failure-induced AFib, heart failure causes fibrosis formation, as you can see in the pictures below. And if the dogs were treated with GGA, no fibrosis was formed. So in this model system, it looks like that GGA protects against fibrosis formation and therefore can prevent atrial fibrillation. A third model in the lab of Natel is the acute ischemia-induced AFib model. If you introduce ischemia in the atria, it causes atrial fibrillation, but when the dogs were pre-treated with GGA, again, we observed the protective effect. In this model, the protective effect is by conservation of the conduction velocity in the atria tissue. Here you see a part of the atria, and the gray area is the ischemic area. So in a normal situation, if you have ischemic event in the atria, the electrical conduction is delayed around the ischemic area. But in case of GGA treatment, the electrical conduction was as in a normal situation, and therefore it protected against AFib. So the conclusions from this part of the talk are that AF results in exhaustion of heat shock protein expression, and genetic and pharmacological induction of the heat shock protein expression conserves cardiomyocyte function and protects against AFib induction and progression. And the heat shock protein inducing compound, GGA, may be an interesting compound to treat clinical AF. And later on in my talk, I will continue on translational research with this compound. The second mechanism we tested in the experimental model systems is the role of epigenetics. And we know that epigenetics is a very important process in switching gene expression on and off. And therefore it has a very important role in cell function. How does it work? In yellow, you see the histones, and around the histones in the nucleus, the DNA is wired. And the histone acetylate transferase will put an acetyl group to the histones, and then the DNA is more loosens up, and then the gene expression can become activated. HDACs remove the acetyl group, and therefore they close gene expression or shut down the gene expression. In the meantime, we know that there are several HDACs, histone deacetylases. They are divided in several subgroups. And we know from already that the first group, the HDAC 1, 2, 3, and 8 are the classical HDACs, so they remove the acetyl group from the histones. The class 2A are not histone deacetylases actually. These histone HDACs bind to pathological transcription factors such as MAP2. And after stress, they are transported out of the nucleus, and therefore the MEF2, for example, can become activated and leads to pathological gene expression. The class 2B group is present in the cytosol, so not in the nucleus, but present in the cytosol. And in the cytosol, it can deacetylate contractile proteins and thereby alter the contractile function of the cardiomyocytes. The HDAC 3 group are the sirtuins, and they have an important role in metabolic homeostasis. And HDAC 11, for this HDAC, the mechanism is not exactly known. So what is happening when you have cardiac stress, such as AFib? HDACs become activated. In the nucleus, the HDAC 1 class can deacetylate the histones and cause pathological gene expression and cardiac remodeling. In the cytosol, the HDACs can deacetylate structural and contractile proteins and lead to contractile dysfunction and cardiac remodeling. And we set out to test various HDAC inhibitors, whether they can prevent cardiac remodeling in experimental AFib setups. So what we did is we used several HDAC inhibitors, so the broad spectrum inhibitors such as TSA, which can inhibit several classes of HDACs, or the sodium butyrate, which is also broad spectrum inhibitor, or more specifically, we use nicotinamide, which blocks HDAC3 class, or tubicin, which is a specific inhibitor of HDAC6. So we treated the cells with the inhibitors. Under normal tachypacing, they had no effect, but after tachypacing, we observed the following findings. Tachypacing causes contractile dysfunction, but when the cells were treated with nicotinamide or tubicin, we observed significant protection against contractile dysfunction. And in the Drosophila, we observed the same findings. Tachypacing resulted in heart dysfunction, but Drosophila treated with nicotinamide or tubicin showed conservation of contractile function of the heart wall. Then we set out to investigate in more detail the role of HDACs. And here too, we prepared several constructs of the HDAC6, because this is such a specific finding, that tubicin, which is a specific blocker of HDAC6, can prevent against AFib. So we investigated in HDAC6. HDAC6 contains two catalytic domains, the histone deacetylase domain and the tubulin deacetylase domain. And we prepared 3 mutant constructs, one with a mutation in the HDAC domain, or in the TDAC domain, or in both domains. We overexpressed the constructs successfully in the HL1 cells. We measured the contractile function on during normal pacing. You see that the contractile function is normal. After tachypacing, we observed again that the control cells showed contractile dysfunction as the HDAC wild-type overexpression cells. But interestingly, when you overexpress HDAC6 constructs with a mutation in the TDAC domain, the tubulin deacetylase domain, we observed conservation of contractile function. And this means that this tubulin deacetylase domain is very important for contractile dysfunction after tachypacing. And interestingly, tubicin, the protective drug, blocks the TDAC domain. So this suggests that the microtubule network may be involved in atrial fibrillation promotion. So we stained the cells for tubulin, acetylated tubulin, on the normal situation and after tachypacing. And we observed that tachypacing causes disruption of the microtubule network. As you can see, here you see a spaghetti-like structure in the cell, which is gone after tachypacing. But when the cells were treated with tubicin, this structure was conserved. We validated the findings also in human tissue. In human tissue, we observed that paroxysmal atrial fibrillation and persistent AFib causes a decrease in tubulin expression, but also a decrease in acetyl tubulin expression. In parallel, we also measured HDAC6 activity, which was increased in the patients with atrial fibrillation, and the expression of the HDAC6 protein was also increased. So these findings are all in line with the findings in the HL1 cells. To test whether HDAC6 is a druggable target, we again contacted the lab of Stanley Natel, and they were willing to test the HDAC6 inhibitor tubistatin A in the dogs. Here you can see that 7 days of atrial tachypacing causes spontaneous atrial fibrillation in the dog. And when the dog was treated with tubistatin, we observed almost 100% protection against AF. We also measured changes in action potential duration, which were prevented by tubistatin. Changes in cell shortening were protected or prevented by tubistatin. And tubistatin prevented the activation of HDAC6. So this part of our talk or of our research shows that tachypacing of the HL1 cardiomyocytes and the Drosophila reveals derailment of proteostasis in terms of activation of HDAC6, degradation of the microtubule network, and contractile dysfunction. And derailment is prevented by an HDAC6 inhibitor. In line patients with paroxysmal and persistent AF reveal loss of tubulin proteostasis and also induction of HDAC6. And we obtained proof of concept for HDAC6 inhibitor in the dog model for AFib. So all the data show that HDAC6 inhibition is a promising target in clinical AF. As I mentioned, also nicotinamide protects against experimental AFib. So we also investigated how is nicotinamide blocking AFib and can it be a druggable target? So what is nicotinamide? Nicotinamide is a form of vitamin B3, and nicotinamide is part of NAD. And NAD is a very important coenzyme found in all living cells. What is NAD doing? It's so important in the metabolism of cardiomyocytes, as NAD is necessary for ATP production. It modulates also various ion channels, such as the sodium channel, but also calcium signaling. And NAD+ is consumed by mediators, including PARP. And now it's becoming interesting because what is PARP? PARP stands for poly-ADP ribose polymerase 1. And this protein is involved in DNA repair. Here you see an overview of the PARP1 gene. It contains a catalytic domain. And when PARP becomes activated, this catalytic domain consumes NAD to make ADP, which is bound to the modification domain of PARP1 to form these strings. And this is called PARylation. So PARylation is necessary for PARP1 to become active and to be active in the repair of DNA damage. We observed that nicotinamide is also, which is also a PARP inhibitor from shown by other studies, protects against contractile dysfunction in the tachyphase HL1 cardiomyocytes. So how can this work then in PARP repair of DNA damage? How does it work? If you have DNA damage, you can see here a single strand break in the DNA, PARP becomes recruited and PARP will bind to the single strand break, and then it consumes NAD and leaves as a rest product nicotinamide and makes these poly-ADP ribose, so the PARylation products. And in this case, PARP is active, it repairs the DNA and the cell can function normally again. So we set out to test, do we see activation of PARP and DNA damage in human atrial tissue? And what we observed in the right atrial appendages and in the left atrial appendages of patients with AF, we can clearly see an increase in the PARylation of proteins in the cell. Interestingly, the PARP expression levels were conserved, but due to the activation of PARylation, we thought, hey, is there DNA damage in the cells? So we measure DNA damage by measuring various endpoints for DNA damage. The first endpoint is the gamma H2AX. We measured the level of positive nuclei for this marker of DNA damage in human tissue. And we observed a significant increase in gamma H2AX in AF versus controls in sinus rhythm. We also measured levels of 53BP1, also a marker for DNA damage, and the nuclear circularity. We observed an increase in the levels of 53BP1, and we observed a reduction in nuclear circularity. And this is exactly what we expect when you have DNA damage. So here we show that patients with atrial fibrillation show DNA damage and an activation of the DNA damage repair pathway via PARP1 activation. As I mentioned, PARP needs NAD to become active. So we tested this whole system of PARP activation in 2 model systems, the HL1 cells and also the isolated red atrial cardiomyocytes. If you tachyphase HL1 of the red atrial cardiomyocytes, we observed a strong increase in gamma H2AX. So this means there is DNA damage in the cells, which is accompanied by higher levels of pyrylation. That is also what you can see here in this Western blot. If you tachyphase the cells, you see more pyrylation, so more a smear of pyrylated proteins. In the same time, we measured the NAD levels, and it's in line. If you have more pyrylation, it consumes NAD, so the NAD levels drop. And this is accompanied by loss of calcium transients and an increase in oxidative protein and DNA damage. And this fuels the process of remodeling. When we block PARP1 with PARP inhibitors or by adding nicotinamide to the cell culture, we prevented all these endpoints. So this shows that if you block PARP1, it's protective against contractile dysfunction. And in this paper, we showed that is due to prevention of NAD loss in the cells, because NAD is so important for the energy consumption, ATP formation in the cells. If I put all the data together, AFib leads to excessive DNA damage, PARP activation, and NAD depletion, which leads to electrophysiological and contractile dysfunction and oxidative protein and DNA damage. And this drives AF promotion. And when we block PARP1 by various compounds, nicotinamide or direct PARP inhibitors such as 3AB, ABT, 888 or olaparib, or when we genetically knock out PARP1, it prevents NAD depletion, it prevents oxidative DNA damage, and it prevents AF promotion. So also PARP1 inhibition with nicotinamide can be an interesting druggable target in clinical AF. So the conclusions from our bench work are that atrial fibrillation is associated with derailment of proteostasis, and key modulators of proteostasis are druggable targets in the preclinical models. So the next step is, can we translate it to the clinic? For example, can these modulators be biomarkers, or can we think of drug studies, which can be beneficial in the end in patients with AF? So this is the second part of my talk, just a few more slides on the translation to the clinic. First of all, we set out to test whether modulators in the pathways can represent biomarkers to stage the severity of AF. Why is this important? As you know, AFib can be detected with an ECG, but you can only detect whether you have AFib or you don't have AFib. You cannot detect the amount of remodeling. So it would be very nice to have a biomarker that can stage the severity of AFib or predict the recurrence of AFib after treatment, or even can predict the onset of atrial fibrillation after a cardiac surgery. So we measured blood-based hetoprotein levels. We measured hetoprotein 27, 70, cardiovascular hetoprotein, hetoprotein 16, but none of them can stage AFib. But HSP 27 and 70 showed some beneficial effect to identify patients with the recurrence of AF after treatment. We observed a significant increase in hetoprotein 27 levels during follow-up of patients who showed a recurrence of AFib. Another biomarker which we tested in serum of patients is mitochondrial DNA levels. And we observed that mitochondrial COX-3 levels and also NAD levels are significantly increased, especially in paroxysmal AF, and become exhausted during persistent, long-standing, persistent AFib. The levels nicely correlate with each other. And the levels were significantly increased in patients showing a recurrence of AF after treatment. So there is maybe some potential for mitochondrial DNA levels in blood as well. And the last marker is the DNA damage marker, the 8-OHDG, in serum of patients. And these levels become increased in patients with persistent AFib. They are increased in patients showing a recurrence of AFib after an ablation technique procedure. And the levels are increased in patients showing a recurrence of or an onset of AF after cardiac surgery. So there's also a potential of this marker as a predictor of the AFib stage and recurrence after treatment. How about the drugs? As I showed in this talk, that GGA is protective against AFib, but also nicotinamide. These compounds are already on the market and in clinical phases for other diseases. So that make the step to a clinical trial in AF patients within reach. The HEAT protein inducer used in many studies is Cellbex, and a nicotinamide is available on the market as Niagen. Together with, as GGA has never been registered in the European Union, it was difficult for us to conduct a GGA trial in the Netherlands. GGA has been registered in Russia, in Japan, China, Taiwan, many Asian countries to treat gastric ulcer. And TEA was available for us to conduct the following trial. We have two patient groups. One group of patients was treated with GGA 3 days before surgery or placebo. And during surgery, the left and right atrial appendages were collected and shipped to Amsterdam for HEAT protein expression level measurements. And Thea mentioned that GGA was well tolerated by the patients. Here we show the findings of the Western blot, and we observed that GGA treatment for 3 days already significantly increased the HEAT protein 27 levels in atrial tissue, and also the HEAT protein 70 levels in tissue. So this was a remarkable finding because I did not expect that 3 days of treatment already can induce HEAT protein levels in atrial tissue. We also observed that GGA, in case of GGA treatment, the HEAT proteins were more likely to bind to the sarcomeric structures. So this gives a hint about the protective function of HEAT protein 27 in the samples. And as all the model systems we use so far were all pre-treatment studies, it is clinically highly relevant to investigate whether GGA can also help in the recovery from damage. And therefore, we developed the following experiment. So we tachypaste the cells for 10 hours and let the cells recover. And during recovery, there was no recovery of the calcium transients in the cells. But when the cells were post-treated with GGA, we observed acceleration of recovery in these cells. And that is what you see here in the quantified data. Here we observed that GGA treatment accelerates recovery of the tubulin network and the acetyltubulin, and it increases the HEAT protein 27 levels in atrial cells. So this is indicating about how GGA can help in the recovery of important proteins for cell function. So the next step we are working on currently is a clinical trial with GGA in the Netherlands. But this is a difficult road and with many hurdles because GGA was not registered so far. And to overcome these hurdles, we founded, like in 2016, a foundation. It's called the Atrial Fibrillation Innovation Platform. And this platform is aiming to collaborate better between patients and research on innovative research in the field of atrial fibrillation. And patients can donate to our foundation to boost the clinical translation of our findings. And we obtained funding from patients, and therefore we can now conduct the Niagen trial in AF patients. And this trial will start this year in the summer. And in this trial, we aim to include 25 patients with heart failure in combination with atrial fibrillation. And these patients all carry an ICD that enables continuous remote monitoring of AFib. And the idea is that we first monitor during a period of 3 months the AF episodes in the patients. So we can, for each individual patients, we will determine the AF burden. And then we will start with Niagen treatment and see for each individual patient whether Niagen can reduce the amount of AF episodes. So we are very happy that patients contributed to this study, and hopefully we will be able to get positive outcomes from this trial. So this brings me to the last slide. I want to thank all the members from my lab who contributed to the studies. Also, I work in close collaboration with Professor Natasja de Groot from the Translational Electrophysiology Department in Erasmus Medical Center. And Natasja is cardiologist and electrophysiologist, and she is helping me with the translation of our findings. And of course, many thanks to the lab of Stanley Natel, who was willing to test several of the compounds in the dog model for AFib. And I want to thank you for your attention, and I'm happy to take any questions.
Thank you, Bianca. Thank you. That was really an excellent overview. And you really, as I expected, you really brought us from the cells to patients. So it is really what we were looking for, a perspective. So I don't know, I'm sure there will be some questions. We have a lot of people connected. Now, I don't know if anyone...
Shall I stop sharing, or do you want me...
It's fine. I think it's fine. I'll break the ice. I mean, before I pass to the next to the my colleague, Professor Charretta, who will make conclusions. I think one of the most interesting aspects of the studies that you showed is that, of course, you didn't, when we look at electrophysiology studies, we are expecting basically electrophysiology studies in cells. But what you showed is basically what really matters is remodeling and what stays behind, and molecular biology. Yeah. What intrigues me is that basically in all your studies, the focus were obviously the atrium and atrial cardiomyocytes, but also many of the pathways that you described are activated in the left ventricle, so in ventricular myocytes. So do you predict that these drugs might be useful also for heart failure, or might be detrimental for some aspects? And also because in most of your models, you looked at atrial fibrillation, but in clinical practice, we see that very often atrial fibrillation is coexistent with heart failure, yeah, or with other clinical conditions. It's not isolated. It's not an isolated condition. It has comorbidities. It has a lot of variables. So I'm wondering whether if you change other comorbidities or other conditions, do you expect to get the same result?
Yeah, that's a very interesting question. I think that definitely part of the pathways, such as heat shock protein exhaustion and also DNA damage, or at least mitochondrial dysfunction and the nicotinamide, can have beneficial effects in heart failure as well. And in America, they are currently testing Niagen in heart failure patients, because heart failure patients, although the mechanism is different, the endpoint is also that there's less ATP production. And therefore, Niagen is currently under a clinical trial in heart failure patients. And for example, GGA in the Netherlands, there's another research group testing or investigating the HFpEF heart failure with preserved ejection fraction, and they found that heat shock proteins have beneficial effects in those patients as well. So together with the HFpEF consortium, we are now teaming up to get GTA registered in the Netherlands to treat HFpEF patients, so a HFpEF trial in parallel with an AFib trial. So yes, indeed, these pathways have common etiologies for heart failure and AFib as well. And we also studied in the past the structural changes in patients with, for example, dilated cardiomyopathy and hypertrophic cardiomyopathy. And this is published in our Nature Review in Cardiology paper 2017. And there I show an EM pictures of ventricular cells and atrial cells. And the phenotype, if you look to the structure of the cardiomyocyte, it's the same. They all show myolysis, so the breakdown of the sarcomeres. They have a hibernative phenotype. So I definitely believe that there are comparable pathways for heart failure and atrial fibrillation.
Excellent. Thank you. I don't know if we have any other questions.
Yes, I'd like to know more precisely which is the aim and the points of the study you announced is starting next summer.
The first endpoint is a reduction in AF episodes, and secondary endpoints will be the effect on heart failure, but also the effect on several biomarkers, the proteostasis markers we measured so far already in serum of AF patients.
So it's a summary in humans of your results, your research, biomarkers and clinical.
Yeah. So in the Niagen study, the first aim is, of course, can this compound have a beneficial effect on AF episodes? So can it reduce the amount of AF episodes? That is a primary endpoint. And secondary is heart failure and biomarkers. Yeah.
Thanks. Congratulations.
Thank you.
So if we, sorry.
Can I ask Dr. Brandele a question? I would like to, can you hear me?
Yes.
Okay, good.
Clear.
Okay, this is Roberto Pedrinelli, and congratulations for your presentation, which is testified by the number of persons connected, which is really a feat. I would like to ask you something which I didn't understand. Do you think that what you have shown in atrial fibrillation is something specific for atrial fibrillation, or is something that is a general mechanism that you find in tachycardia-induced cardiomyopathies?
Whether the mechanisms will be the same, nobody knows, but I think there's some overlap. But during atrial fibrillation, you have a very high activation rate of the atrial cardiomyocytes. And that will trigger different pathways, to my opinion, than if you have heart failure in ventricular cardiomyocytes. And so the trigger is different. But for example, heat shock proteins, they have a very general cardioprotective effect by binding to several binding partners in the cardiomyocytes. So the small heat shock proteins tend to bind to the sarcomeres and protect them, for example, against breakdown. So whether you have heart failure in the cardiomyocytes or whether you have tachypacing and induced stress in the atrial cardiomyocytes, these heat shock proteins will protect the sarcomeric structure. And as if by these EM pictures I mentioned, we clearly observe that patients with cardiomyopathy have myolysis, as well as patients with AFib. So, and it can be that the heat shock proteins can have a beneficial effect, even if the triggers for the stress are different.
Okay.
But you know, this is just all, although we observe these phenotypes in human atrial tissue, and although heat shock proteins also protect against heart failure, there are many studies showing this. It has never been compared in clinical trials. So that is, I think, very interesting for the future to conduct clinical trials with GGA. In our case, in AF patients, but also in the HFpEF patients, and see whether we find comparable pathways involved. This has never been studied so far.
Yes, thank you. Thank you very much. And congratulations again.
Thank you.
So if we don't have any further questions, I will call Sebastiano.
Yes, I'm here. Can you hear me?
Yeah, for the conclusions.
Yes, conclusion, and let me ask also some questions because the talk was very interesting. And before I start my small comment, let me congratulate Dr. Brundel for this great talk. And I think it was really terrific, and I'm really proud that this talk was organized on behalf of our working group. And I also thank Dr. Indolfi, Mercuro, and Petrinelli for the coordination of the society that allowed us to organize this talk. And my small comment is just related to the fact that, in my opinion, the topic of your presentation is really important because, of course, this was a perfect example of translational research that starts from an important unresolved question from the clinical and brings it to the basic levels in order to find new therapeutic targets for the treatment of the disease. And I think that we are doing this for most of cardiovascular diseases, such as myocardial ischemia, heart failure. And if you think about heart failure treatment, many of the drugs that we are using now in the clinical practice targets specifically the pathophysiology of this disease. On the other hand, I think there is a huge gap between the mechanism underlying atrial fibrillation and the drugs that we use in the clinical practice. Because basically in the clinical practice, we are not targeting any of the pathophysiological mechanism underlying the development of this arrhythmia, whereas we just practice rate or rhythm control and anticoagulation. I think it's really needed a research like yours that highlights new targets that are able to directly intervene on the mechanism underlying the disease so that we can prevent it and maybe avoid the progression. And my questions are basically oriented on this aspect. So you elegantly showed many mechanisms behind the progression of atrial remodeling that, as you know, are basically the basis for the development, for the transition from paroxysmal atrial fibrillation to persistent and permanent. And I wonder whether all these mechanisms also play a role in the mechanism triggering the beginning of atrial fibrillation at a molecular level. You know that triggers, re-entries, ectopies are all electrophysiological triggers that promote the development of atrial fibrillation. So do you think that HSP27 or epigenetic modification targets, for example, channels of proteins involved in calcium handling or potassium transitions or depolarization? So this is the first question. And the second question, you know, I'm from Rome, right? So as all the roads lead to Rome, I think that in your presentation, all roads lead to autophagy, basically, because you know, nicotinamide, HDAC inhibitors are potent activators of autophagy, and you know, mitochondrial dysfunction and accumulation of mitochondria in your model, it may suggest, as you said, an exhaustion of mitophagy mechanism, and you know, also autophagy is really involved in elimination of protein aggregates. So, what do you think about these two important aspects? And thank you again.
Thank you. For the first question, do you think whether I think that the pathways may prevent the triggers? Of course, we don't know, but the clinical trial will learn, because we need to get more insights in whether these pathways are also involved in the recovery. So far, we tested 2 compounds in post-treatment models. One of them was a GGA, which accelerates the recovery from cells. And the other compound was the PARP inhibitor. We also tested in a Nature Communication paper as a compound that can also stimulate the recovery from damage. So these are 2 very promising compounds, and the clinical trial will teach us whether it can help in the repair. And of course, when you do clinical trial, we cannot measure the repair on the structural level in the heart of patients, but we hope to see this repair in a normalization of the biomarkers. So certain biomarkers go up when you have AFib, and maybe with the treatments, these biomarkers like the DNA damage marker or the mitochondrial DNA biomarker, hopefully they go and normalize again. So that is an indirect suggestion that we will have recovery of the atrial tissue. For the second question related to autophagy, autophagy is very important for normal physiological cell function. And therefore, we hypothesized that it would not be beneficial to directly interfere with the autophagy pathway. What we did was we interfered upstream of autophagy by blocking ER stress with heat shock proteins. And by blocking upstream trigger for downstream excessive autophagy, we prevented AF remodeling in HL1 cells in Drosophila and even in the dog model for AFib. Because of the length of this talk, I could not include all those data from that study, but it has been published in the American Heart Association in 2017. So I think for the autophagy, it's more the upstream target, which we can modulate with drugs. I hope this answers your question.
Oh, perfect. So I want to leave to Cinzia, Mercuro, Perinelli, and Indolfi closing remarks. Okay.
Yeah. Thank you.
Thank you, Bianca. Thank you again. Really, it is exactly what we were looking for. We were looking for a perspective that was from basic to clinical. And that's the aim of our working group. And also, even in the ESC, we have the same aspiration to start from basic insights and get into the clinical scenario. So thank you very much for this excellent perspective. Yeah. I don't know if Professor Indolfi can or Professor Mercuro want to add anything, or Pedrinelli to close the meeting.
For us and for me at least, is a lovely, exciting experience. I hope you will continue. This goal is perfect for us, for our society. Thank you very much. Your role.
Thank you for the kind invitation. Also, Cinzia, thank you for inviting me.
Yeah, you were voted. You were the one that got more votes. So I proposed, but you were the most voted one. So
if you are very much interested in AFib, this week Nature will put our paper on AFib online tomorrow.
Oh, great.
If you want to get the whole holistic concept of AFib, then you can read this paper.
Thank you very much, Bianca. Thank you very much. And talk to you soon.
Yeah, bye-bye.
Good afternoon, everyone. Bye-bye. Thank you for attending the meeting. Bye-bye.
Yeah, bye.
Bye-bye. Thank you.